Good morning, everyone, and thank you to the organizers, thank you, for inviting me to speak about converting variance to function. And I thought I'm going to walk you through one of our recent GWAS dissection loci and give you kind of a high-level overview of that because of time restrictions, such that we can take that to the discussion and talk about challenges and opportunities to accelerate and facilitate that effort. So, no. Okay, sorry. Yeah, certainly not a lot to add in terms of GWAS discoveries, speaking after Eric and Mark, but the bottom line is that GWAS have been incredibly successful in identifying over 100,000 genetic associations that are robustly associated with diverse traits and diseases in humans. And that is kind of in sharp contrast to the fact that only a handful of those have been converted to actionable therapeutic hypotheses. So the question is, why is that the case? And I'm coming back to our work on the, and I apologize, there's some rearrangements in the slides. They should look more structured. I don't know what's wrong here, but I'm just going to continue, hoping that most will be good to read. So in our work on the strongest genetic association with obesity, the FTO obesity risk locus, we kind of defined a set of 6 key questions that we consider is important to tackle when converting variance to function. And this includes, Eric has mentioned it, and Mark as well, to ask which of the associated variants are actually the causal ones, in which cell type and context do they act, and how do they affect upstream regulators and downstream target genes, and what are the cellular and organismal phenotypic consequences of those. So another layer to add in here is that there's growing evidence in the community that actually many of these loci confer pleiotropic risk and associate with multiple phenotypes at once, such that studying these pleiotropic effects might actually be a great opportunity to discover variants and mechanisms that are shared between these traits. And in that context, we were intrigued by a locus on chromosome 3 that Mark has mentioned briefly before, and that we find confers an increased risk for type 2 diabetes and hyperglycemia that has been reported in the past. And I will come back to that later. Fantastic work has been done by Anna Gloin, Mark McCarthy, Guy Rudder, and others in the community. But we found that this locus actually confers not only risk to hyperglycemia and type 2 diabetes, but also to low bone mineral density. And in the following slides, I'm about to tell you about our work that took in total almost 6 years, where we convert a regulatory variant on the one side that changes the chromatin structure by affecting the binding of a regulator, stereoresponse element binding protein 1, and affects ADCY5 expression in adipocytes and osteoblasts, and has an impact on cellular phenotypes that are relevant to the organismal traits, bone mineral density and hyperglycemia, which is namely, on the one side, an increased stress-induced lipolysis rate in adipocytes, and on the other, we find and implicate for the first time that osteoblasts actually depend on fatty acid oxidation early on in differentiation processes in order to go through terminal differentiation, synthesize collagen, and build the bone matrix. So taking a step back, here's our association signal, which is located and contained in the first intron of ADCY5 and harbors 13 variants in high linkage disequilibrium. And we first asked, so what is the cell type and tissue involved here? And we looked at chromatin state reference maps from the Roadmap Epigenomics Consortia and the ONCODE Consortium. Anshul is here, the lead author of that effort. And we find that this locus is actually spanned by a polycomb repressed chromatin structure, specifically in the mesenchyme. And data that I don't show you here is that we find a particular enrichment of the histone mark for polycomb repression, which is H3K27ME3, in adipocytes and osteoblasts. So we evaluated these cell types further, and for that we built, not only for this work, but also for others, a biobank of adipose-derived mesenchymal stem cells, or short AMSCs, that we can differentiate into adipocytes and osteoblasts. In doing so, we tested if the haplotype has actually differences in terms of chromatin accessibility in our AMSC cells that we carry through throughout differentiation by an assay that tests for open chromatin accessibility, a TAC-seq. And we find indeed that haplotype 1 is enriched for open chromatin by twofold compared to haplotype 2. So we asked, what is the variant driving this difference in chromatin accessibility? And we used orthogonal computational approaches, some of which we have developed in the past. Actually, on the left side, you see a deep convolutional neural net trained on genome-wide chromatin accessibility data in our AMSC cells throughout differentiation. And these data show or prioritize one variant, RS56371916, to increase chromatin accessibility for haplotype 1 compared to haplotype 2. And then we turned to sequence-based conservation analysis and predict the binding regulator, stereo-response element binding protein 1, which then in a series of experiments that I cannot share today, but we validated the effect both of the variant as well of the regulator. And we asked, what is the downstream target gene here? Or are there several downstream target genes? And we looked at three-dimensional genome folding maps in mesenchymal stem cells and find that this locus lies in a very well-defined contact domain, only implicating two genes, which is ADCY5 and PTP-LB. And we perform a haplotype-specific gene expression analysis in our AMSC cells and differentiate them to adipocytes and osteoblasts, and can say that there's a clear EQTL in those three different cell types, in these three different cell types for ADCY5 expression, comparing heterozygous with homozygous individuals. I will note here that we did not detect any EQTL at the tissue level, so neither adipose tissue nor the bone showed us an EQTL. Coming back to tissue is heterogeneous. We need good reference maps in the right cell type and in the right context. So that brings us to a model where the variant basically shifts the dynamic balance between poised chromatin, which is both accessible as well as repressed, to repressed chromatin by affecting the binding of stereo-response element binding protein 1, thereby changing ADCY5 gene expression in adipocytes and osteoblasts. And as a last experimental slide, I want to show you our work where we wanted to confirm that the haplotype effect on ADCY5 expression as well as on cellular phenotypes is actually driven, at least partially, by this particular variant. And we did so by performing CRISPR genome editing to make isogenic changes at this particular SNP in AMSC cells, and then we send them into differentiation to osteoblasts and adipocytes. And we can not only show that the variant edit actually leads to a rescue of ADCY5 expression. You don't see these data here. What you see is that the variant edit actually leads to changes and has an impact on cellular phenotypic signatures that are relevant to bone mineral density and type 2 diabetes. Namely here, osteoblast marker gene expression and catecholamine-stimulated lipolysis rate. So I've mentioned before, this work took in total almost 6 years, and I will come back to the work of many colleagues in the field that have specifically looked at another partially overlapping haplotype, which associate with type 2 diabetes and high fasting glucose. And this haplotype, or these groups, particularly here, Anna and Mark, and also others in the field, have implicated beta cell function for ADCY5 in the context of type 2 diabetes, which to me, and we will have great discussion, we already said we would discuss, it's a fantastic, interesting target, ADCY5, in the context of pleiotropy, I think. So overall, and this took almost 6 years, this work, and I will say that in 2012, when we started, it wasn't easy to get Jiva summary statistics from the bone consortia, so we needed time for that. That's much better in place now, which is fantastic. We also needed to generate our own reference maps at certain cell stages, and throughout differentiation of these cells, we had to set up a biobank. All that takes time, and looking forward into the next phase, as Eric has opened it, I think things are getting into place, and collaborating is here really the key. Collaboration is truly a key in this story here. 5 different labs with diverse expertise, with access to patients, with access to Jiva summary statistics. We're working closely together over these years. And one on the experimental side, I want to stress, and that's what my lab is mostly thinking about over the last 2 to 3 years, how can we scale up these efforts? One example, the CRISPR variant editing experiment took 4 years until we had isogenic changes in osteoblasts and adipocytes. Mark, isn't it? It's an insane timeline that we had to go through and that everyone has in the field, such that we are truly focusing right now in other labs as well to scale that experimental approach and to get it to a point that we can edit hundreds of variants in a parallel fashion and then train the right computational models to actually predict the effects in a much better way. Another thing that we are thinking about a lot is how can we preempt the effect of genetic variants on molecular and cellular phenotypes. And here I will say that we have a fantastic, very close collaboration with Cecilia Lindgren, who's in the room and then has organized the meeting. And we've built a biobank of AMSC cells from 500 individuals, 200 of which we have visceral and subcutaneous adipocytes, and we have immortalized control cell lines that we now plan to carry through high-content cellular imaging as well as molecular profiling and live cell imaging to connect genetic variation on the one side to cellular phenotypes without a priori assumption, and then linking those to diseases and phenotypes. And the way we started out is that we now compiled and aggregated millions of variants to genome-wide polygenic scores for metabolic traits, and that we select and distinguish particularly those individuals that are at the tails of the genome-wide polygenic score to send them then into these cellular and molecular profiling assays. So I think my time is over. I want to thank my lab. My lab has been fantastic over the last years. And my collaborators, I'm only mentioning those here that were involved in this project. A big thank you goes to Eric. It has been fantastic to work with you on the ADCY5 work, as well as with many other colleagues in the field, Douglas Keel. The work with Cecilia is fantastic, and I'm very happy to take questions and discuss later in the breakout session. Thank you very much.